Good morning, everybody. Today's, as we discussed in the last class about the taste, which is also called as gustation. Today's class is about sense of smell, which is olfaction. And as you all know, it is most primitive of all our senses. It is also called as a chemical sense. It is highly developed in animals, so it's called as macroosmotic, whereas in humans it's highly developed, so it's called as microosmotic. Physiologically, taste and smell are called as visceral senses because they have close association with GIT. The flavors of different foods items are a combination of gustation and olfaction. The importance of smell is it is helpful in nutrition. It increases the gastrointestinal secretion and motility. It helps in defense. It is also helpful in the animals among animals for environmental hazards. It helps in reproduction, like the male gypsy moth can be attracted by a female at a distance of 3 miles. And one important, the olfactory memory cannot be decayed. It is the longest memory, whereas the pleasurable olfactory memories are very resistant to decay and retain lifelong. The emotional tie between the mother and baby cannot be retained. It is always retained lifelong. Sorry. The uniqueness in olfaction or olfactory sensation is the receptor cells are exposed to the external environment, which are bipolar cells are called as telereceptors. The olfactory receptors undergo a rapid turnover, so they can regenerate. They have no direct relay to the thalamus. The fibers from the primary olfactory cortex pass to the dorsomedial and are concerned with discrimination of odors. They don't have any neocortical projection. So there is no relay station in the thalamus. The olfactory apparatus, which encompasses the olfactory epithelium or the mucous membrane, is located in the only place where the nervous system is very close to the external world. It is located in a specialized portion of mucous membrane, that is the yellowish pigmented olfactory mucous membrane, which is around 2 to 5 square centimeter. It is larger in lower animals. The aeration or the ventilation is poor along the olfactory mucosa. The air reaching the olfactory mucosa is greatly increased by sniffing. The olfactory receptor cell is a bipolar cell which lies between the supporting cells. There are about 10 to 20 million receptor cells. Each has a cell body, an axon, and a dendrite. The dendrite is a short, thick, which extends to the free surface of the olfactory epithelium, where it forms the olfactory rod. Here you can see in this diagram the olfactory rod and the cilia, which is exposed to the outer surface, and olfactory epithelium where it expands to the olfactory rod and which bears the olfactory cilia. There are about 10 to 20 cilia for each rod. The cilia are unmyelinated and the cilia contain odorant receptors of smell. There are more than 1000 different types of odorant receptor proteins. The axons of the bipolar cells are unmyelinated and they pierce the cribriform plate and enters the olfactory bulb, where they form the first order neuron. That means the first order neurons end in the olfactory glomeruli. You can see the axons piercing the cribriform plate of ethmoid bone. The supporting cells, which are also present, are columnar irregular. They separate from the bipolar neurons, and these supporting cells are similar to the glial cells, and there are basal cells, which are the source of receptor cells. The mucus is produced by the Bowman's gland present below the basal lamina of the olfactory membrane. The mucus contains the antibodies, the odorant binding protein, and the enzymes. The antibodies prevent the entry of bacteria, whereas the odorant binding proteins facilitates the movement of odorant towards the receptor. The enzymes helps in removing the odorants. So we came across the different supporting cells in the progenitor cells, which are essential for the regeneration. The receptors are short, thickened dendrites and expanded olfactory rod, and the rods from the rod cilia project to the surface. There are 10 to 20 cilia per neuron. The axons of the neuron pierce the cribriform plate of ethmoid, which ends in the olfactory bulb, where the lifespan of the olfactory receptor is around 2 months, and it is replaced by the basal cells. The supporting cells end in the microvilli, which secrete mucus and Bowman's. Here you can see the superior turbinate, the bluish discoloration, which shows the olfactory mucosa also spreading towards the septum. The sense of smell is perceived along the chemoreceptor, which is sensed by the olfactory cilia, and it passes through the olfactory bulb. From there it continues as olfactory tract or the olfactory nerves, and it gets integrated in the amygdala, hippocampus and olfactory nucleus. So this is the place where the first order neuron, that is the axons of the bipolar cells, pierce the cribriform plate of ethmoid, and they synapse with the mitral, tufted and granule cells, where the olfactory bulb is formed. The olfactory bulb is a flattened ovoid body seen in the cribriform plate of ethmoid. The different cells of the olfactory bulb are the mitral, tufted, granule and periglomerular. The axons of the bipolar cells synapse with the dendrites of mitral cells to form complex glomerular synapses called as olfactory glomeruli. About 1000 olfactory neurons synapse on the dendrites of a single mitral cell. The tufted cells are small triangular cells whose dendrites form the olfactory glomeruli. The mitral and tufted cells form the second order neuron of olfactory pathway. The granule cells are inhibitory, they have no axons. The dendrites of the mitral and tufted cells, they synapse with and they form dendrodendritic reciprocal synapses, which do not follow the one-way conduction as it follows in all the synaptic connections, and the neurotransmitter is the GABA, the gamma amino butyric acid. There is also periglomerular short axon cells which sharpens and focuses the olfactory signals. There is a clear-cut regional projection from the olfactory epithelium towards the nervous system. That is, the upper part are projected to the upper part of the olfactory bulb, and the lower part of the neurons are projected to the lower part. That means the upper part of the superior turbinate projects to the upper part of the olfactory bulb, whereas the lower part projects to the lower part. So this is how the sense of smell. You can see here how within 2 months the olfactory epithelium is replaced. And here you can see how it pierces the cribriform plate of ethmoid, forming the glomeruli, that is, the axons of the bipolar cells and the dendrites of mitral and tufted cell form a glomerular synapse, whereas you can see the mitral cells continues as the axons to the second order neuron. The olfactory receptor cells, as you all know, synapse with the mitral cells. The particles which reach the olfactory epithelium from inspired air should be volatile and lipid soluble. Highly lipid soluble substances have strong odors. Substances with same number of carbon atoms but different structures will have different odors. Humans can distinguish around 10,000 different odors. The receptor proteins are located on the cilia of the bipolar cells. The receptors are chemo and telereceptors. The first order neurons are the bipolar cells. Axon, the bipolar pierces the cribriform plate and reach the olfactory bulb, where it synapses with the dendrites of mitral and tufted cell to form the second order neuron. The synapse forms the olfactory glomeruli. The axons of the mitral and tufted cells pass posterior as the olfactory tract. As they reach the anterior perforated substance, they divide into three striae: the medial, the intermediate, and the lateral striae. The medial striae is less distinct in humans. It is formed mostly by axons of the tufted cells. They cross to the opposite side in the anterior commissure and reach the olfactory bulb and synapse in the granule cells. That means they cross the midline. They are contralateral side, whereas the lateral and olfactory are ipsilateral. That means they pass to the same side of the tract towards the olfactory cortex. The lateral striae is formed by the axons of the mitral cells, whereas the intermediate striae is formed by the rest of the axons of mitral cells, which they go to the olfactory cortex. The olfactory cortex includes the anterior olfactory nucleus, the prepiriform cortex, the olfactory tubercle, the amygdala, the limbic system, the septal nuclei. As you all see, the olfactory axons bipolar pierce the cribriform plate. They end in the synapse of the axons in the glomeruli with the dendrites of mitral and tufted cells, and they send axons. They form the olfactory tract and they reach the cortex. Here you can see how it is piercing the cribriform plate of ethmoid along with the olfactory epithelium in the superior turbinate. The olfactory cells are periglomerular and granular cells, which we discussed. The granular cells are inhibitory, whereas the periglomerular cells are sharpening and focusing the odor, in discriminating the odor. The granule cells have the receptor neurotransmitter called as GABA. The axons of mitral and tufted cells continue as intermediate and lateral striae, whereas the axons of tufted cells continues as medial striae, which crosses to the opposite side. Here you can see in the cortex, the amygdala is responsible for emotional responses, whereas the entorhinal cortex is the olfactory memory. So once they reach the olfactory cortex, the preamygdaloid area, the piriform cortex and prepiriform cortex of the cerebral cortex are known as primary olfactory cortex, whereas the entorhinal cortex of the parahippocampal gyrus, which is area number 28, is forming the secondary olfactory cortex. Here in this diagram, you can see how the epithelium pierces the cribriform plate, where it synapses with the dendrites of the mitral and tufted cells, and the axons of the mitral and tufted cells continue as olfactory tract, which can be medial, intermediate, and lateral. The lateral and intermediate pass along the same side, whereas the medial olfactory striae crosses over, that is, it decussates to the opposite side and synapses with the granule cells in the opposite side. How is the processing of olfactory information takes place? It is in the prepiriform cortex that more complex aspects of olfaction, that means the integration takes place in the primary olfactory cortex, which is the prepiriform cortex. The entorhinal cortex is concerned with olfactory memory. From the entorhinal cortex, fibers are projected to hippocampus, insula, orbitofrontal cortex, which are the areas of olfactory discrimination. The orbitofrontal cortex or part of neocortex is concerned with conscious discrimination of odors. To be very precise, the integration takes place in the primary olfactory cortex, whereas the secondary olfactory cortex is concerned with the olfactory memory, whereas the discrimination is by the hippocampus and orbitofrontal cortex. The amygdala and hypothalamus are concerned with emotional and behavioral responses. If the anterior commissure is sectioned or cut, the olfaction of both the sides are impaired, whereas if the olfactory tract is section, the olfaction of the opposite side is cut. A newly phylogenetically olfactory pathway has been the one which passes through the dorsomedial nucleus of thalamus and terminates in the posterolateral quadrant of orbitofrontal cortex. This pathway is concerned with conscious perception of odor. There are some inhibitory pathways which are called the cortico-pugal inhibitory pathways, which pass from olfactory regions of the brain backward to the olfactory tract and terminate in the specialized inhibitory cells called as granule cells. The olfactory transduction or the stimulation of the olfactory cells or the propagation of impulses takes place when the smell of volatile substances or bind to the odorant binding protein. See, here you can see how the odorant chemical combines with the odorant binding protein. It gets activated because of the stimulation of adenyl cyclase, and it is a G protein coupled receptor, and where the cyclic AMP is formed from CAMP, cyclic AMP is formed from ATP. Here the membrane potential of the unstimulated olfactory cell is about -55. It activates the gated sodium channels. The sodium ions enter through the gate, which is open. Stimulation of the odorant substances reduces the potential from -55 to -30, and the entry of sodium ions depolarizes the cell membrane, wherein it transmits the action potential, which is propagated along the olfactory nerve to the CNS. The receptor cells generate action potential at a very slow rate. The rate varies proportional to the logarithm of stimulus intensity. So it is a G protein coupled receptor which activates the adenyl cyclase, which forms the cyclic AMP, which opens the sodium channel, depolarizing the membrane. So this is how the sodium influx causes depolarization. The depolarization of the olfactory receptor cell membrane triggers action potential in the action. So what is olfactory threshold? The threshold varies for different substances which comes in contact with epithelium, and they get dissolved in the mucous layer. It is measured with an instrument called as olfactometer, also called as Zwardy Marker's olfactometer. The olfactory threshold of methyl mercaptan in garlic is 1 per million of a milligram per liter of air, and for chloroform it is 3.3. This relevance is methyl mercaptan is usually mixed with natural gas to detect leak from pipeline. The sensitivity of different substances are discriminated remarkably, and humans can perceive around 10,000 different odors with a threshold change of around 30%. That means the concentration of around 30% should be changed in a molecule and an odor to be appreciated. The odor producing substances are small, 3 to 4 to 8 to 20 carbon atoms. The molecules come in contact with the receptor cells, and they generate a potential which are graded potential and result in activation of the cyclic AMP with reduction in membrane potential and opening of sodium channels, causing action potentials to be propagated along the axons to the olfactory bulb. There is an individual variation in response, and it is initiated by odorant binding proteins. The olfactory adaptation or the olfactory pathway shows adaptation. Initially it's clear odor and gradually it decreases. That is, as you go into a toilet, initially you start singing, it is stinking, later you get adapted to that. It is mainly by changes in the receptor, or it can be a central phenomenon. Adaptation is specific for particular odor. The central phenomena due to changes occurring in So it can be due to central phenomena or changes occurring in the receptor. 50% is mainly due to receptor change. The central phenomena is due to strong inhibitory feedback to olfactory bulb from olfactory cortex. There is also something called as masking of odor. An offensive odor can be masked by a pleasant odor. We all wear deodorants, so this is the relevance. The principle is made use in perfume industries. The deodorants do not remove the offensive odor, they only mask it with pleasant odor. So, what are the abnormalities in? So, we have anosmia, absence of smell, it occurs in common cold, fracture of cribriform plate, or mechanical obstruction. Parosmia is altered in the sense of smell, which occurs in sinusitis. Or hyposmia is decreased sensitivity, which happens in hypogonadism, or hyperosmia due to hysteria, which is increased sensitivity, and neurasthenia. The threshold or increases with advanced age. That means the olfactory sensitivity decreases with advanced age. Olfactory hallucinations are perceptions of non-existing odors occurring as an aura to epilepsy or secondary to temporal lobe lesion. An aura is defined as a movement, a sensation, or a mental disturbance which precedes an epileptic convulsion. Females are more sensitive to smell, especially at the time of ovulation. So today's class we came across what is the peculiarities in olfaction, what is the olfactory pathway, and how does the signal transduction takes place. That means how is the processing or how do you perceive the smell. So we have an odorant binding protein which initiates the conversion of or activation of adenyl cyclase, opening the sodium channel with reduction in membrane potential, and the action potential is generated and transmitted along the olfactory pathway. You know how is the olfactory pathway formed? It is formed by the axons of the axons of the bipolar neurons, which pierces the cribriform plate, synapses of the dendrites of mitral and tufted cell, and that is the first order neuron. The second order neuron continues with the axons of mitral and tufted cells forming the medial lateral intermediate striae. The medial striae crosses through the opposite side and synapses of the granule cells and projects onto the primary and secondary olfactory cortex, whereas the intermediate and the lateral, the synapse on the same side, that is the ipsilateral. And you came across the words like adaptation and masking. The questions will be describe the olfactory pathway or what is an olfactory receptor and the abnormalities. So we will discuss tomorrow in the web section. Thank you. Stop it.